Good morning, it's July 2nd, and this is your daily brief in science. Here's everything you need to know. A new study from the University of Surrey reveals intriguing insights into how brain stimulation can enhance learning in mathematics. Led by neuroscientist Roy Cohen-Kadosh, the research found that participants with weaker connections between the dorsolateral prefrontal cortex and posterior parietal cortex exhibited improved calculation learning after receiving electrical stimulation to the DLPFC. The study involves 72 students from the University of Oxford and aims to optimize learning while addressing cognitive disparities. It emphasizes the significant role neurobiology plays in educational outcomes, suggesting that biological factors may outweigh environmental influences in math learning. This aligns with the Matthew effect, where individuals with early educational advantages continue to excel. Cohen-Kadosh highlighted the potential of this research to assist those struggling with learning, but also raised ethical concerns regarding accessibility and the risk of widening socioeconomic gaps. Researchers stressed the need for scientifically validating consumer devices based on these findings to ensure their efficacy and safety. However, there are limitations to the study, including a small sample size and an average participant age of 21, which may not translate to children. Improvements were mainly seen in response speed rather than accuracy, which could affect the practical implications in educational settings. The research utilized a multimodal approach, taking into account behavioral performance, brain connectivity, and individual differences among participants. Future research is needed to explore the connections between neurochemistry, brain plasticity, and the effectiveness of neurostimulation in real-world educational contexts. Cohen-Kadosh expressed optimism about the potential for at-home brain stimulation technology, especially for individuals with learning difficulties, though previous studies indicate that such stimulation could negatively affect those with high math skills. Researchers at NYU Tandon School of Engineering, led by Weiqiang Chen, have unveiled a new device called the Leukemia-on-a-Chip. This innovative technology, roughly the size of a microscope slide, integrates the physical structure of bone marrow with a functioning human immune system, offering a promising avenue for testing blood cancer treatments. The Leukemia-on-a-Chip allows for real-time observation of how immunotherapy drugs interact with cancer cells in a controlled environment that closely mimics human physiology. This aligns with the FDA's ongoing push for alternative testing methods, particularly as conventional methods often fall short. Animal models do not accurately represent human immune responses, and laboratory tests fail to capture the complex interactions occurring within the body. The timing of this development is critical, as the FDA is moving towards phasing out animal testing for specific drugs, encouraging innovations like the Leukemia-on-a-Chip. The device aims to enhance treatment outcomes for patients undergoing chimeric antigen receptor T-cell therapy, a promising but challenging treatment for blood cancers with a high relapse rate and serious side effects. This new technology can simulate various clinical scenarios, including complete remission and treatment resistance, and evaluates the efficacy of newer fourth-generation CAR T-cells compared to standard versions. Researchers have also created a matrix-based analytical index to assess different CAR T-cell therapies, aiming to tailor treatment approaches to individual patients. By utilizing advanced imaging techniques, the team observed immune cells actively hunting and destroying cancer cells, revealing purposeful movement patterns that were challenging to study in living systems before. The leukemia chip recreates the essential regions of bone marrow where leukemia develops, allowing for self-organization and retention of the immune environment, potentially revolutionizing how blood cancers are treated. Recent research from Weill Cornell Medicine has unveiled a significant shift in our understanding of how the brain generates energy. Published on July 1, 2025, the study reveals that brain cells can utilize fat droplets as an energy source, challenging the long-held belief that glucose is the sole energy provider for the brain. This groundbreaking research opens new avenues for exploring the connection between lipids and neurodegenerative diseases. It suggests that fatty acids derived from lipid droplets may help sustain brain energy levels during fluctuations in glucose availability. In experiments, researchers blocked the enzyme carnitine palmitoyltransferase 1, or CPT1, which is essential for transporting fatty acids into mitochondria. This intervention induced a hibernation-like state in mice, reinforcing the idea that the brain can rely on lipid droplets for energy. The study found that electrical activity in neurons drives the consumption of fat droplets. Active neurons metabolize fats, while resting neurons do not engage in this process. Additionally, experiments demonstrated that neurons can convert triglycerides from lipid droplets into fatty acids, which are then utilized in mitochondria to produce ATP, the energy currency of cells. Interestingly, the research also focused on the DDHD2 gene, which encodes an enzyme responsible for breaking down fats. Mutations in this gene are linked to hereditary spastic paraplegia, leading to neurological symptoms. In mice lacking the DDHD2 enzyme, lipid droplets accumulate, allowing neurons to convert these fats into fatty acids for energy production, especially when glucose is scarce. Principal investigator Dr. Timothy A. Ryan and lead author Dr. Mukesh Kumar emphasized the importance of lipid metabolism in brain energy supply, drawing parallels to its role in muscle tissues. Dr. Kumar noted that these findings align with the metabolic demands of the brain, similar to other active tissues like muscles. Further investigation is necessary to fully understand the connections between lipid accumulation in neurons and conditions such as Parkinson's disease, as well as the interplay between glucose and lipids in brain metabolism. The Nurses' Health Study, which began in 1976, has become a cornerstone of long-term research into women's health and nutrition, involving nearly 300,000 women. This extensive study has significantly advanced our understanding of various health issues affecting women, with participants like Patricia Chubb and her mother, Charlotte May Rohrbaugh, highlighting its critical role. Their involvement underscores a unique aspect of the study: multi-generational participation, where families continue to contribute to this vital research. However, the future of the Nurses' Health Study is under threat due to funding cuts announced in May 2025. Researchers are now facing the urgent task of preserving essential biological samples collected over decades, including urine and DNA specimens. Participants have expressed concern and frustration regarding the current administration's Make America Healthy Again initiative, which they view as ironic given the cuts to crucial research funding that contradict public health objectives. The potential loss of this data has been described as akin to burning the Library of Congress, emphasizing the irretrievable nature of the research contributions made over the years. Political tensions, particularly threats from the Trump administration to cut federal funding to Harvard over alleged civil rights violations, further jeopardize the continuation of the study. This funding uncertainty could hinder the collection of health data from approximately 280,000 nurses, which has been invaluable in understanding women's health. The Nurses' Health Study has produced significant public health insights, including the harmful effects of trans fats and the link between smoking and heart disease. Its findings have influenced national dietary guidelines, hormonal therapies for breast cancer, and research on dietary factors affecting disease. Overall, this study has been unique in its focus on women's health, an area historically underrepresented in medical research. Recent advancements in breast cancer research were prominently featured at the 2025 American Society of Clinical Oncology meeting, with a strong emphasis on antibody-drug conjugates, HER2-positive treatments, and hormone receptor-positive cancer therapies. Among the key studies presented, the ASCENT-04 trial reported encouraging results for the combination of sacituzumab govitecan, also known as Trodelvy, and pembrolizumab, or Keytruda, as a frontline treatment for triple-negative breast cancer. This trial demonstrated a significant improvement in progression-free survival for patients receiving this antibody-drug conjugate compared to those undergoing standard chemotherapy. Notably, the ASCENT-04 trial did not report any new toxicities, aligning with the established safety profiles of the treatments involved. Furthermore, trial organizers allowed patients in the control arm to access sacituzumab after disease progression, reflecting a commitment to patient care. Dr. Kevin Kalinske from the Winship Cancer Institute highlighted additional significant trials, including DESTINY Breast 09 and SERENA-6. DESTINY Breast09 evaluated the effectiveness of TDXD, or trastuzumab deruxtecan, combined with pertuzumab against the established Cleopatra regimen in HER2 positive breast cancer. The SERENA-6 trial focused on switching from aromatase inhibitors to camizestrant upon the detection of ESR1 mutations, which showed prolonged progression-free survival but raised concerns about the trial's design. This trial also examined the clinical utility of ctDNA testing in frontline hormone receptor-positive breast cancer, introducing new considerations for treatment strategies. Additionally, the inavolisib trial reported improved overall survival in high-risk patients with hormone-refractory disease treated with a triplet regimen, although there are ongoing concerns regarding toxicity management. The updates from ASCO indicate promising advancements for breast cancer patients, underscoring the need for continued research to optimize treatment strategies. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.